Hello, everyone, and welcome to today's webinar, Transforming Integrated Solution. I am Cassie Soltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Galatech. To learn more, visit galatech.ai. We encourage you to participate today by submitting any questions you may have during the presentation. Participants in the live broadcast will have the opportunity to receive a free demo of Galatech's Liquid Workstation or Organoid culture kits. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's webinar. Professor Hanry Yu, Professor at the Department of Physiology, National University of Singapore, Institute of Digital Medicine, Translational Research Program, NUS Medicine. Today, Dr. Zheng will participate in the Q&A session to address questions on behalf of Professor Yu. Dr. Yicheng Liu, Senior BD Director at Galatech, and joining us for the Q&A session is Dr. Yu's colleague, Dr. Kuxiao Zheng. Dr. Liu, you may now begin.
Hello everyone, today is really my pleasure to join this webinar to introduce the organoid research and also drug discovery solutions from Galatech. And in the beginning, I just want to give you a brief introduction about our company so that you know what is the current development status of our company, what we are doing and who we are. And our company is actually established in the year of 2016, and we specifically providing advanced tools and automations for life science, healthcare, and also drug discovery. And as you can see on this map, the global headquarters of our company is in Singapore, and globally we also have presence in Japan and also in the US. And recently we also registered a legal entity in Munich, Germany. And in the future, we want to build up the R&D center, the customers experience center, and also our operational office. And the majority part of our company is based in China, and the headquarters of our China division is actually in Suzhou. And within China, we have R&D center, lab, delivery center, and also factories all over China. And the mission of our company is to empower digital transformation with advanced production tools to make the world healthier and better. So since our company looks at technology evolution and also product development very importantly, so this is the reason that we establish the collaborationship with many famous international and Chinese universities. And some key facts about our company. So at this moment, we have employee more than 1,000, and more than 60% of our employees have master or above degree. And at this moment, we already applied for more than 1,000 patents. From all this number, you can see this company is already well established, and we are also a totally scientific, technology-driven company. And due to many years successful business operation, we established a good relationship with many key customers, for example, Agilent, Danach, J&J, Novo Nordic, Wuxi Pack, PJ Group, and many Chinese and also international companies. In the meantime, we also attract the attention from many investors, and some of you may notice the investors are also our customers. And this is a perfect demonstration to showcase the trust and also win-win situation that we established with our customers. So actually, we are at the beginning phase of our business operation in Europe. We just entered into the European market by the end of last year. So for the initial phase, we will specifically focus on life science and healthcare. And under this business sector, we have standardized routine instruments. We also can provide the integrated solutions. So when talking about the standard routine instruments, we have liquid handling robots, we have microplate handler, we have nucleic acid extractor, and we can also provide high content imaging as well as many other routinely used lab automations. So because of the time reason, I'm not able to introduce the details for individual instruments. However, today I will highlight a few key instruments. The first automation that I want to highlight today is the liquid handling workstation. And our liquid handling workstation is an open platform. So when I say open platform, it means the reagents and consumables from any suppliers could be used on our platform. And additionally, this platform is also equipped with easy-to-use software. So for example, I don't have any bioinformatics background. However, I only need 2 days to master all the key functions on this software. And furthermore, this platform is also equipped with cutting-edge technology. As you can see on this video, we have 4 or 8 independent pipetting channels. It means the pipetting channels can work totally separately. So we can adjust the space and also the height between individual pipetting channels. And in addition, we also have the liquid level detector, the bubble sensor, and also the cloud sensor. And furthermore, this instrument also can automatically adjust the pipetting speed according to different liquids. And last but not least, I want to emphasize the capability from our company to provide the customized solution. Because we are a manufacturer, we can integrate many different functional modulars, for example, microplate reader, centrifuge, RT-PCR, PCR instruments, and also cooling modular, magnetic stand, and many other functional modulars on this platform. And because of the time reason, I'm not able to finish the whole video. So in the next part of my presentation, I want to highlight the high content imaging. So this machine is widely used for high throughput drug screening, and it features 5 color fluorescence channel, large field of view, and also fast imaging. We only need 0.6 seconds to take a picture. And we can also provide the software solutions so the end users can do multi-field imaging stitching, cell segmentation, 3D reconstruction, and also biomarker analysis, as well as many different functional units. Most importantly, we can also integrate the high content imaging into the microplate handler so that we can build up a small integrated platform. So in the second part of my presentation, I will specifically highlight this one-step automation. And the core capability of our company is to provide integrated solutions. This is also how we distinguish ourselves from the other automation suppliers. We can provide not only small robotic units, but also we can offer the hardware, software, and also our expertise to build up integrated solutions. As you can see on this slide, we can offer high throughput drug screening system, the genomics system, cell line development system, and of course also the organoid culture and analysis system. And this is also the solution that we want to highlight in the next part of my presentation. The reason that we can build up such complex integrated solution is because we can provide 5 layers different support. First of all, we can provide the state-of-art instruments, for example, the liquid handling workstation and also the high content imaging that I mentioned in the previous slides. Additionally, we can also provide software which can seamlessly connect all the instruments, and by using our software, we can also set up the experiments schedule the experiments, and also design the protocols for individual instruments. And additionally, we can also provide a software which can do data analysis, archiving, storage, and also generate the report. And in the future, on the top of these existing software solutions, we also want to provide artificial intelligence program so that our platform can do independent machine learning, data analysis, and also decision making. In the next couple of slides, I want to summarize the mission and also our perspective for the future life science infrastructure. Within our company, we think the life science infrastructure has gone through 4 different revolutions, from traditional manual lab to standalone automation to pipeline automation. And we think in the future, the next wave of life science infrastructure revolution is powered by the artificial intelligence combined with lab automation. So within our company, we want to provide a dry lab and wet lab combined solution so that by using our platform, people can do not only experiment designing, producing data, but also collect, storage, analyze, and archiving the data. And if we want to provide this dry and wet lab combined solution, we must be able to provide state-of-art automation. We must be able to provide software to improve informatization and also digitalization. On the top of this, we also want to provide artificial intelligence program so that we can further improve the data quality, reducing the running cost, increase the working efficiency, and also improve the data security. That brings me to the end of my first part presentation. In the second part, I will specifically introduce the Organoid solutions from our company. And as we know, the Organoid culture is extremely cumbersome, and it includes sample collection, sample preparation, centrifuge, Organoid seeding, culture, exchange the medium, and also many steps for drug treatment and also imaging data acquisition. And currently, the lab technicians and scientists are suffering from non-standardization, low data consistency, and also low sample throughput. This all because of the cumbersome Organoid culture procedure and also the many manual steps within the Organoid culture protocols. And within our company, we can provide multi-level Organoid automations. For example, the standalone automation, the one-step integrated solution to high-level integrated solution. In the next part, I will first introduce the standalone automation. And this platform is transformed from our standardized liquid handling robots. And we specifically integrate the temperature control and cooling modular, tilted modular, and also centrifuge so that our Organoid workstation can do Organoid seeding, centrifuge, and also exchange the medium. And for the upgraded version, we can also do the we also integrate the 96 pipetting channel and also the tilted modular so that our platform can do also the drug treatment. And here I just want to quickly summarize the key advantages of our Organoid workstation. So first of all, our Organoid workstation has comprehensive function. It can do Organoid seeding, centrifuge, exchange the medium, drug treatment. And secondly, this platform is also equipped with easy-to-use software. It has very intuitive interface and user-friendly setup. And additionally, we also have very strong capability to fulfill the customized requirements. So for example, we can integrate many different functional modulars, and we can also provide various carriers for different consumables. Last but not least, we also provide multiple self-developed culture kits and all these kits are already validated on our Organoid workstation. In the next part, I will introduce the one-step automations based on the smart plate handling system from our company. So first of all, a quick introduction about the smart plate handler system. So our system could maximum integrate small robotic units, and it's a benchtop platform. It has very compact designing, and additionally, we also put a full axis robotic arm on this platform so that the robotic arm can do lid opening, barcode scanning, and also transport the sample plates from the sample hotel to the instruments. And additionally, this platform also is well designed with high precision and also stable performance. The first one-step automation that I want to introduce is the imaging combined with our smart plate handler. And by using this one-step automation, we can do the Organoid imaging and also dynamic detection. So here, I just want to show you video so that you have further impression about our high content imaging and our smart plate handler and also the one-step automation that I just introduced. As you can see here, this is a very typical working scenario. In the lab, the technicians have to put the samples into the machine one by one. However, by using our robotic platform, the robotic arm can transport the sample plate from the sample hotel to the analyzed instruments. So our high content imaging can do 3D scanning and also live cell imaging. After the data acquisition, the robotic arm transfer the sample back to the sample hotel. And actually, this is the old video, so we still have the old designing on in the video. So in the future, we are going to put the collaborative arm into the platform so that we don't have to use the safety button. And our software also can do cell cycle analysis, cell proliferation, biomarker analysis. This is a side-by-side comparison between manual operation and also fully automated system. The second one-step automation that I want to introduce is the nanoliter dispenser combined with smart plate handling system. And by using this system, we can build up a fully automated system for Organoid dosing and drug treatment. Here, just want to show you a small video, a short video, so that you have further impression about our instrument. So after the drug dispensation, the robotic arm first transfer the sample plate to the sample hotel to close the lid. And then we transfer the sample plate back to the sample hotel. And here we can also put a cell culture incubator. And then the robotic arm can either orderly or randomly grab another sample plate to the instrument. So in the next part of my presentation, I want to specifically introduce the Organoid integrated solution. And for the basic, let me just turn on the laser pointer first. So for the basic level of our integrated solution, we integrate the liquid handling workstation, consumable stack, centrifuge, cell imaging instrument, and also the cell incubator. And the experiment steps that we can cover include organoid seeding, culture, centrifuge, exchange the medium, and also imaging data acquisition. And for the upgraded level of the integrated solution, we integrate liquid handling workstation, cell culture incubator, high content imaging, nanoliter dispenser, centrifuge, cell counter, film sealing instrument, microplate reader, and also the robotic arm. So all the individual instruments could be connected through the robotic arm. And the experiment steps that we can cover include organoid seeding, culture, centrifuge, exchange medium, drug treatment, and also imaging data acquisition. This is a showcase data to demonstrate the organoid automation within our lab. So the throughput of this platform is 42 96 well plates per run. And as you can see, in 30 seconds, we can put the matrigel in the plate. And besides the standardized integrated solution, we can also offer the customized solutions. For example, this is a real customer case, and they want to build up a platform for organoid drug sensitivity detection, and the instruments that they want to integrate in the platform are robotic arm, dispensing system, microplate reader, decapper robot, nanoliter dispenser, and also centrifuge. And these are two pictures to showcase the platform that we build up for the customers. And because if you want to build up the integrated solution, the software is very important. So here I just want to quickly highlight the key functions of our software. So our software is called Lapilin. It has altogether 8 different functional units. And by using our software, we can seamlessly connect all individual instruments on the platform. We can also schedule the experiments. We can set up the protocols for all the machines, and we can also do the um, data management. For example, analyze the data, archiving, storage data, and also generate the data report. And last but not least, so by using our software, the scientists could also monitor the workflow on the integrated platform. And at this moment, our software is powering the cell drug screening platform, the molecular biology platform, and also the protein detected evolution platform, and of course, our organoid culture and analysis platform. In the last part of my presentation, I want to highlight the organoid culture kit. And the designing of our culture kit is powered by the artificial intelligence program. So we first use the artificial intelligence program to do the literature scanning so that we can absorb the information from hundreds and thousands literatures. And afterwards, the program can automatically give the scientists a few recommendations according to the organoid tab and also the tissue tab. And then we can practice all these formulas on our fully automated platform, and then the artificial intelligence program can do the self-independent data analysis. For example, to analyze the organoid imaging data, the biomarker information, the morphological information. And then, according to the data analysis results, the artificial intelligence program will give the scientists some recommendations and suggestions to further optimize the formula, and then we finalize the formula for organoid culture kit. And the advantages of our kits are, first of all, we have strict quality control. We guarantee there's no any contamination from microbiomes. And secondly, our culture kit has very good consistency. This is because the huge production capability from our company. And additionally, by using our kit, we can also guarantee the high physiological relevance. So our kit can return the tissue and genetic characteristics. We can also maintain the primary tissue heterogeneity. Here, I just want to give you some showcase pictures, you can see this is the gastric cancer organoid, and here this is the colon cancer organoid. And by using our kit, we can improve the successful rate of the organoid construction from less than 40% to 90%. And here are just some biomarker informations from our liver tumor organoids. And as you can see, the Ki67, the AFP, the HNF4A, and also GPC3, when you side by side to compare the biomarker expression between liver tumor organoid and also patient's tissue, you can see very high physiological and also pathological relevance. And at the end of my presentation, I just want to quickly highlight the advantages of our organoid solutions. First of all, we can provide multi-level automated solution from standalone to integrated platforms. And we can also provide AI-powered software so that we can do the self-independent experiment designing, data analysis, and also data management. Last but not least, we can also provide total solutions for organoid application. For example, we can provide automations, hardware, software, consumables, and also reagents. That brings me to the end of my presentation. Thank you very much for your attention.
Thank you, Dr. Liu. We will now hear from
for hosting today's talk. Institution listed here, University of Singapore and Mechanobiology Institute, Singapore, for supporting my work. The title of my talk is Current Trends and Challenges in Organoid Research and its Application in Drug Discovery. Is the 3D cell-based construct or models that is complex in vitro models. Also illustrated here on the left, spheroid hydrogel-based scaffold, template tissue engineering construct, bioprinted tissue construct, organs on chip or microphysiological system, and organoid research has attracted a lot of attention and rapidly year after year. 3D constructs from different cell sources, embryonic stem tissue, adult stem cells, or iPS impotent stem cells, or from adult tissues. Dissociated, sometimes in the case of iPS, reprogrammed, under differentiation, molecules, they form organoids under specific or other types of microenvironment. The different cell-derived organoid, such as iPS, generous mature functions remain as a progenitor state. Adult stem cell derived organoids or tumor cell derived organoid tend to show more mature phenotypes. Applications of organoids, such as those listed here: precision medicine, regenerative medicine, toxicology, disease modeling, development, omics, host microbes interaction, etc. More time focusing on the relevance to drug discovery process, specifically looking at the efficacy, maybe in some cases toxicity studies for approval of the drugs and biologics. Rate discovery, tested, evaluated for efficacy, and various stages of clinical trials for safety and efficacy. Clinical stages despite proven efficacy and safety in animal models. Only about 5% of animal tested drugs obtain regulatory approval for human applications. Do this fail? Interspecies differences between human and animals underestimated because biological studies sharing similar functional modules in humans. but the exact components actually varies. 2006. 1912 is an antibody designed to stimulate CD28 and modulate immune activities. 6. Phase 1. It elicited a severe cytokine release syndrome in 6 healthy volunteers, leading to serious adverse events, including multi-organ failure. All the previous preclinical studies of TGN-1412 reveal potential risk because of the animals is very different from human biology. As well as academia has been talking about revamping for 3 or 4 decades. About going to an industry conference, 2D to 3D culture. Companies are skeptical, but they are hopeful because the potential for in vitro human cell-based drug discovery process is so attractive. The promise recapitulate human physiology, physiological complexities, low cost, and various other aspects such as ease of systems and maintenance and such to all and accelerate the drug discovery process. But it has been very until more recently. Reason between how sophisticated or how complex the model need to be versus the robustness of the systems. We've seen adoption of organoid or organ-on-a-chip microsystems by players and even regulatory bodies in the pharmaceutical sector. So some key milestones which is the current state of excitement research for use in drug discovery process. Those milestones by industry and academia, the enactment of the FDA Modernization Act 2.0 in 2022, passed by U.S. Senate, authorizes use of alternative methods, new alternative methods or NAMs, potentially, and this includes cell-based assays and computer models, obtain an exemption from FDA to investigate safety and effectiveness of a drug, removes the requirement to use animal studies as a part of a process to obtain a license for a biological product that is biointerchangeable with another biological product. Would a candidate of a company got IND without using animal studies, but using organ-on-a-chip? Since the FDA Modernization Act 2.0, number of clinical trials of organoid, but trying to, so we call organoid-based clinical trials registered in clinical trial database 2023 and 2024. You see the number keeps on growing. Diagnostics for potential use in drug discovery of organoid. It comes back to the question of how complex the model need to be to reach the physiology of human and the disease context. It is easy for this model to have consistent performance in drug testing applications. For most of the biologists, as complex as possible, as biomimicry as possible, years. Making a very complex model is you can potentially make a huge with no use. When in the terminology of systems engineering, is a measure of uncertainty. One least desired feature of a drug testing assay is uncertainty. So therefore, of various systems, such as the component composition of the cells, the environment it's in, the parameters controlling morphogenesis to generate architecture, etc. need to be very finely balanced. Simple 3D model microphysiological systems. More and more later on. Organoid or 3D in vitro models obviously is important. Here, in figures on the left, culture as 2D monolayer shows negligible morphological changes in response to growth factor stimulation, but becomes invasive if embedded in 3D hydrogel after donation. Picture on panel B, drug treatment only observed in 3D culture as well. 3D does have its significance. You venture from a sheet of cells into the third dimension generates a lot of complexity, which is uncertainty. Manage the third dimension of our research and many people's research. Embedded in the dome-shaped matrix or other hydrogels shows on the right hand side inside the harder it is to control the homogeneity, heterogeneity, balance, ability primer to work in this area. Various inference of structures and preserved integrity and balance between the complexity and homogeneity. Some of them are listed here. The nutrient diffusion limitations, localization. There's one more thing. In many cases, innovation, information in in vitro models. Limited lifespans, limited size because of diffusion limit, heterogeneity, scalability, and standardizations are some of the key problems. The 3D cell culture, complex in vitro models, organoid are facing. Give a little bit more example about physical cues today. To help relieve the heterogeneity or variability issues. Better and more consistent approach is try to engineer something which is more consistent with the help of microfabrication and biomedical to control the consistency of cells when it goes through tissue morphogenesis. Is the input to generate a better paper strain, the stiffness patients to guide the formation of the tissue. Degree of success. Dissect the process of neural tube formation dimension by splitting the first two dimension and the third dimension in a micro-patented substrate. As the form. In the first two dimension, the group has been able to generate neural tube. Once you go to tube. So, but the approach is a wonderful approach. The third dimension formation control. And it has been worked by using a stretched to dynamics the mechanical response to morphogenesis. Earlier, vascularization of keloid by flow will enhance the formation. Peek deeper into how the third dimension. Basically, before we use the third dimension for 1 to 2 cell layer. We really know whether they are in a 3D or 2D. So i.e., the third dimension, a cake, advantages, cell layers, nutrient, drug, waste removal, and perform image analysis and a ball. We and others have been cell sheets. In fact, there's a large group of Japanese for the past 30 years have been working on cell sheets engineering, can be combined together with organoid research. On the left-hand side was effort from us to ligand conjugate onto substrate. So therefore we can cells push to force the cell to stand up. When you're standing up, the cells have a more lateral interaction contact with each other, facilitate the tighter cell-cell interaction. The approach on the right here is trying to membrane, porous membrane, to inhibit the third dimension growth. So therefore, in the presence of fluid flow, cell-cell interaction can be enhanced and functionality can be enhanced. Develop for complex 3D in vitro model such as organoid, in drug discovery and development, to be robust, reliable, and to be sure and with the right control, with operating procedure clearly defined, and vivo and clinical results to validate the performance of efficacy, in some cases the toxicity, to be established. Activity, specificity, a cutoff value of assay, and such purposes requirements. In vitro models such as organoid is ability and heterogeneity arising from both intrinsic and extrinsic factors. Spatial organization of the cells in the construct and the gradients of factors, variability due to the accumulation of metabolites in the environment for the system even in the baseline without any drug introduction. So it's very difficult to find be easily adopted. To monitor and control such variability and heterogeneity to some extent, I've talked about the output side from various groups. I haven't talked about the control from monitoring the variabilities from the output side using label-free methods and imaging methods over time. Side and output side, for example, from input side, as we see from three-dimensional space into the first two dimensions, the steps involved in the culture process increase. The steps of the processing in cell culture also increases the variability and heterogeneity while trying to reduce it. And such an increase in the steps of the processing in cell culture requires automation, precision monitoring, in which out to tell and monitor the functions and behaviors, so therefore you can control the process of cell culture. System or the culture are quite small as a container, so therefore the number of cells and the media ratio is relatively similar to 1:1 or 1:few. And any changes of accumulation of metabolites in the cells and in the organoid will drastically change the environment. Perfusion model has been used, but in many cases, perfusion model has its own drawback. Kind of monitoring requiring that adopted and to monitor the functions of cells and have adaptive adjustment of the input conditions to compensate for the changes of the cell functions over time. If you really want to system for step, but is moving fast towards near forward. Even more complex augmentation of different cell types to organoid structure, example augmentation, vascular augmentation, neuronal augmentation, stromal cell augmentation, and ECM augmentation, especially for cancer-associated fibroblasts. Really topic for research. For inter-organ interaction, organ on chips, microfluidics and such, microphysiological system has become helpful. To solve the problem if the model gets more complex, goes up tremendously. But you need to preserve the associated with approaches. One prominent approach is using each chip, organ on chip. So organoid on chip. So therefore, you can model multi-organ interaction. Simulate multi-cell has become popular in the last few years is assembloid. Right. Assembloid, two kind of tissue from two different regions, or the brain, for example, and form connections when you put them together in the same environment. Aggregate to assemble into assembloid. Approaches is getting more complex. Suggestion has been complexity. Environment, investigate this more focused. A lot of imaging, a lot of data analysis. So AI seems to be playing an important role. Advanced 3D and 4D imaging and laboratory automation seems to be very important in this process as you dissect the process in a very complex organoid construct. On the left-hand side, since 2018, 17, 18 or so, people start creating chip. This is one approach. The panel on the right is assembloid tool. This balance between complexity and variability. I am proposing to be complex, focus on a very well-defined function of interest to keep the function of interest simple. Some of the example here is underlying here. Be complex. The model can be complex. Degrees of ability. Focus function of interest need to be very well engineered, with the rest of the complex tissue less stringent. How the organoid model becomes increased of interest in an ever complex model remains well dissected, well defined. FOI is a reduced model with I haven't touched upon various aspects of an AI-based readout control to establish baseline. Those controls can involve sensors, real time, so to also help resolve the dilemma between complexity and. Let's leave it here. Thank you for your attention. Hopefully this will be useful and it will be followed up with a dialogue about automation of cell culture systems. Thank you for your attention. Bye-bye.
Thank you for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type the question into the Ask a Question box and click Submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started here. Our first question asks, how is sterility achieved during the processes?
I guess this is the question for me, and I want to take the opportunity to answer this question. So actually, the answer is very straightforward. So when we build up the integrated solution, we can also put the HEPA system on the system so that we can maintain a clean environment. And additionally, we can also separate different functional units or compartments. So for example, we can separate the DNA, RNA extraction unit from the RT-PCR instruments. So I think through this kind of way, we can also prevent the cross-contamination from happening. Yeah, that's my answer for this question. Thank you.
Wonderful, thank you. Okay, moving on to our next question. This question asks, what do you consider to be the primary technical challenges in organoid culture, and how does Galatech address these challenges?
And as I mentioned in the webinar, so the biggest challenges are no standardization, low consistency, and also the low throughput. These are caused by different skills of the technicians, too many manual steps, and also complex cultural processes. So Galatech tackles all these problems by offering multi-level automations from standalone to integrated solutions so that we can improve the standardization, consistency, throughput, and also working efficiency. Yeah, I think that's the answer for this question. Thank you.
Excellent, thank you. All right, our next question here is for Dr. Zheng. Is automation worth the investment for academic organoid labs?
Oh, I'd say that is a very interesting and a very good question because there will be when we're talking about automation options, there will be expectations, and there will be concerns. There will be also there are uncertainties about risks, and if the benefits outperform the cost. I think that really depends on the skill and the your requirement, your task, specific task of the research work in the laboratory. And I think it is more realistic because, as I think I recall, one of the Dr. Liu's slide just showed that there is a series of different level of automation from entry level to medium to large workstation or a whole package system. So the realistic solution might be that the core facility of the department or a school to to procure this medium or even larger system to accommodate multiple research groups to share the resources, and they can also assign the core facility technicians, managers to take care of these complex auto systems. And on the other hand, I think for individual research groups, maybe it is time to consider that to use some sort of modular standalone automation system to replace the tedious and error-prone steps during the whole workflow. For example, the daily cell culture medium exchange. Actually, in Dr. Yu's lab here, we are trying to implement sort of this kind of automation system to remove some of these time-consuming and nobody like steps in there. Yeah, thanks.
Okay, thank you. Our next question that we have here says, what are Galatech's primary advantages in providing automation solutions related to organoids except different levels of automation?
Actually, this is very good question. I think after my presentation, the audience would understand that we try to provide different level automations to reduce the burden of the lab technicians and also scientists. In addition, we also want to make the working process more repeatable, consistent, and also standardized. So most importantly, we have a strong capability to fulfill the customized requests. We can integrate different functional modulars or small robotic units in our standalone or integrated platform. And as I mentioned within my presentation, on the top of all these advantages, we can also offer a total solution, including hardware, software, reagents, and also consumables. Thank you.
Okay, wonderful. Thank you. We have time for maybe one more question. This next question says, given that artificial intelligence is currently a key area of focus, could you elaborate on how Galatech integrates its AI technology with automation solutions?
Okay, this is another very good question. So I can probably give you 2 examples. And one of the examples that I already mentioned in the presentation. So the designing of our Organoid culture kits. So as I mentioned in the webinar, so we use the AI program, conducts the literature scanning, and design the the formula of the culture kits. And afterwards, we also practice the formula on our fully automated platform. And after data acquisition, the AI program also does self-independent data analysis and also gives the suggestions to the scientists for how to optimize the formula. And the second example that I can think about is the applied chemistry. So our AI program can predict the affinity between small moleculars and also the protein targets, and synthesize the small molecular and also test the function of the drug through our fully automated platform. And at the end of the data analysis, the AI program can also inform the scientists how to optimize the structure of the drug candidates. Yeah, these are 2 examples that I want to highlight today within the webinar, how we use the AI program to to make the life of the scientists easier. Yeah. Thank you very much. That's my answer.
Thank you. And we have one that just came in that we might want to talk on. This question states, do you know about NASA using microgravity and their simulated rotational microgravity on Earth? These achieve 3-dimensional organoid growth, including co-culture, to achieve extreme high-fidelity tissue-like structure. NASA automated this for Earth and space station research.
Yeah,
I
speaking
actually
of,
don't know this news. Do you know anything about this, Dr. Zheng? This is
yeah,
actually very
actually, this
interesting
is,
information.
yeah, this is very interesting. People are thinking about the space to bring organoids into space. Actually, speaking of NASA, one colleague of Professor Yu in the Microbiology Institute of NUS, they also collaborated with the same organization, I mean NASA, 2 years back. They have fabricated some, specially designed microwave plate for, microgravity cell culture applications, and they didn't bring that to space station, but they did some microgravity, in-flight test. That's a very interesting direction. I think, especially personally, I would say we should maybe pay attention to muscle research in space, as recently, just yesterday, the United States, they just brought back their astronauts, being there for 9 months or so, and they can't even stand up. So I think organoid research, and especially automated solutions, would really pay off for these efforts of space physiology, because if you can put automation there, then you can lower the astronauts you need to put in the space station, right? Yeah, thanks.
Wonderful. I want to thank you both for your time today and your important research. And we would like to thank LabRoots and our sponsor, Galatech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we do not have time for today, and those submitted during the on-demand period will be addressed via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and goodbye.
Thank you. Goodbye.